I'm going to be reviewing the process of translation. Translation involves decoding mRNA into amino acids that are then able to be linked together to form a polypeptide chain, with the end product being a protein. These proteins are needed for the survival of the organism. mRNA consists of RNA nucleotides that together form a genetic code. There are 4 different RNA nucleotides: adenine, uracil, cytosine, and guanine. Adenine and uracil both bond with each other, and so they are considered complementary, while cytosine and guanine bond with each other, and so they are considered complementary. 3 of any one of these RNA nucleotides in a sequence on an mRNA strand form a triplet code that expresses an amino acid. There are 20 amino acids that make up functional proteins. The number of possible combinations of 3 nucleotides is 4 to the third, which is equal to 64. The genetic code is considered to be degenerative, since an amino acid can be expressed by more than one series of nucleotides. For example, the codon GGA and GGG both code for the same amino acid, which is glycine. Additionally, the genetic code is unambiguous since only one amino acid is expressed by the sequence of 3 nucleotides. 3 nucleotides on mRNA form a codon. All codons code for an amino acid except for 3. They are called stop codons or termination codons. They do not code for the amino acid and mark the end for protein synthesis. The start codon, also known as the initiation codon, marks the beginning point for translation. It codes for the amino acid methionine. RNA nucleotides are added to the 3' end of the mRNA strand. Therefore, it is written in the 5 to 3' direction. Translation has 3 stages: initiation, elongation, and termination. After mRNA undergoes post-transcriptional processing inside the nucleus, it leaves the nucleus via the nuclear pore and ends up in the cytosol. At the initiation stage, initiation factors help attach the 5' end of the mRNA strand to the small subunit ribosome. A ribosome consists of protein as well as ribosomal RNA and is made inside the nucleolus. Now, it's important to understand the structure of a transfer RNA molecule, also known as tRNA. On one end, tRNA contains the amino acid that codes for the codon on the mRNA strand. For example, CUC codes for the amino acid leucine, which is why this transfer RNA molecule contains the amino acid leucine. The other end of the tRNA consists of an anticodon, which is complementary to the mRNA codon. However, wobble pairing can occur at the 3rd nucleotide position, which means that the 3rd nucleotide has some flexibility with bonding. For example, UCU can attach to the AGG anticodon. After the mRNA is attached to the small subunit ribosome, tRNA containing the anticodon UAC obtains the amino acid methionine and attaches to the complementary codon sequence on mRNA at the P site. This causes a large subunit ribosome to help assemble the initiation complex. The E, P, and A site on the ribosome contains 3 tRNA binding sites. The A site is known as the acceptor site, which receives tRNA molecules with their amino acids attached. The P site is the site in between the A and the E site. The E site is known as the exit site, where the tRNA molecules are removed. During elongation, a tRNA molecule attaches to the A site that contains the anticodon and the amino acid for the mRNA codon. The carboxyl end of methionine, which is at the P site, attaches to the mean end of the amino acid on the A site. This is done using ribosomal RNA-based peptidyl transferase, which catalyzes the reaction. This is an example of a ribozyme, RNA molecule capable of catalyzing specific chemical reactions. The amino acid attached to tRNA at the A site ends up being the second amino acid, while methionine is going to be the first. The ribosome then moves one codon over in the 5' to 3' direction, where tRNA binds to the anticodon to the mRNA codon. When this occurs, the tRNA molecule that was at the P site is now on the E site, while the tRNA on the A site ends up being on the P site. The A site once again becomes available to receive a new tRNA molecule. As elongation continues, the polypeptide chain continues to grow until it reaches a stop codon at the P site. Note that as a polypeptide chain is formed, folding occurs based on the sequences of amino acids with the help of proteins called chaperones. During termination, a stop codon, also known as a nonsense codon, attaches to the mRNA strand at the A site. Release factors bind to the A site, which allow water to be added to the end of the polypeptide chain. The polypeptide is then disassociated from the tRNA and the ribosome. The ribosomes separate into subunits where it will be reused for protein synthesis.